food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Ptáček, thank you for joining me.
My pleasure.
It's a bit fortuitous that we're talking now because we've had this on the books for a little while. We're going to talk about circadian rhythms and how they change and how they are regulated. And I just got off a plane last night from the East Coast back to my home in Seattle. So I'm in the middle of starting the shift back. I was there for like a week, so I had just sort of adjusted, and then it was time to come home. But do you want to start off by telling everyone a little bit about you and what you study?
Yeah, so I'm a neurologist by training, and I've always been interested in genetics and began my career, scientific career, using human genetics, studying families initially with one rare inherited disease where I went on to map and clone the gene. And so we've worked on trying to understand the genetics of many different hereditary neurologic diseases. But along the way, I became interested in normal sleep, and that was because of families that we identified that have genetic sleep traits that are not diseases, but that transmit in the families. And the beauty of that for someone like me as a human geneticist is that if we have families and we get DNA from the families and we characterize them clinically, then we can use the DNA to map and clone genes, identify genes and mutations that cause whatever trait it is we're studying, a neuromuscular disease and a form of familial epilepsy, for example, or most recently, in the case of sleep, families with segregating traits for being extreme morning larks. So these people are healthy and happy otherwise, but they're incredibly early morning larks, and they're that way from early childhood all the way through elderly life.
And so, what can you tell us about just? I want to give everyone an overview before we jump into the new paper and some things about how various environmental signals interact with the core circadian clock of the body. I want to give people a sense for the basic logic at the molecular genetic level for how a clock even exists inside of us. So a lot of people will know who are listening to this podcast that there are circadian rhythms. We've got these rhythms in our body that help us, regulate our sleep-wake cycles and our behavior that are somehow synced to the external world. The sun rises, the sun sets, all of that stuff connects. But I want to give people a sense for the oscillations that happen at a molecular level. So can you start to describe at a high level, at a basic level, the transcriptional loop that is just constantly on inside of mammalian cells and how that works?
Right. If I could go back even a little further than that, I like to think of everything in the context of evolution. So our planet is circling on its axis every 24 hours, and Darwin predicted that there was selection for people, or for organisms, for any life form, selection for those that can survive and propagate in the environment. And for us, that means a 24-hour day. And so all of our physiologies have evolved, and we have evolved with our physiologies being regulated in a circadian way. And if we survive, that means that we're doing well. We have the advantage of surviving and procreating, propagating our genes. So being in sync with the circadian rhythms is critical to our survival and propagation as a species. So what we learned initially from Drosophila genetics, and then also some mouse genetics, and then our work in human genetics, is that there's a feedback loop, a transcriptional translational feedback loop, where a couple of genes and proteins are turning on other genes that then are translated to proteins that can or not be, may or may not be phosphorylated, which affects their stability. And those proteins then can translate back into the nucleus, where they turn off the transcription of their own genes. So circadian proteins are turning on two circadian genes or multiple circadian genes in us that make protein that feeds back to turn off the transcription of their own genes. And then those proteins have a certain half-life and can be degraded. And once they are degraded to low enough levels, the whole cycle turns on again. So it takes, for a cell, one of our cells, a skin cell, fibroblast, for example, in culture, we can show that the process of those circadian genes being turned on, translating to proteins, translocating to the nucleus, and then being degraded as proteins over time, that whole process takes approximately 24 hours. And circadian comes from the Latin circa, approximately, about, dian, which is a day, about a day. And so that whole molecular feedback loop requires about 24 hours. And then, because we have to live on a day length that's exactly 24 hours, there have evolved mechanisms for tweaking the clock to make sure that we're on a 24-hour day and not a 24.2-hour day.
I see. So it's not, so there's this auto-regulatory transcription loop in our cells. Basically, some of the core proteins here are transcription factors, proteins that bind to DNA and turn on and turn off other genes. So you've got some proteins, they get transcribed, they get made, they start turning on a bunch of other genes. That includes genes for other transcription factors that actually turn off the first gene. And there's this loop of activity. One gene or one protein turns on a bunch of genes, including a gene that'll encode a protein that'll eventually turn off the first protein. And there's this loop that happens. So things are being turned on, they turn other things on, those things turn the first things off, and so on and so forth. And you said that there's a degradation time constant here. The proteins only stick around for so long. And when you study in detail the mechanics and the dynamics of this whole loop lasts for about 24 hours. Not exactly 24 hours, but about 24 hours. And I think an important thing I want to ask you about is, so this loop is just sort of intrinsically inside of our cells. It's set up to be a 24-hour period, but it's not inherently tied to daylight or nighttime or anything. It has to be sort of locked into being in sync with the external world. And so there has to be some kind of communication between the outside and the inside so that this periodic transcription protein loop can be synchronized to the external world.
Right. And it's probably, certainly very, very difficult to imagine, and perhaps impossible, for us to develop in our cells a clock that ticks at exactly 24 hours per day. Mm-hmm. So we humans, on average, have a clock, a circadian period of approximately 24.2 hours, plus or minus 5 or 6 minutes. Mice, like the mice we study in the lab, C57 black 6 mice, have a period that's slightly shorter than 24 hours, 23.7 hours. So they're very close to those times, 23.7 for that strain of mouse, 24.2 for us humans. But then it's the light at the beginning and end of the day, each day, that says, because our periods as humans is longer than 24 hours, it's the light in the morning that's so important. It says, hey, dummy, you shouldn't be sleeping. You need to get up because the light's out.
Right.
The sun is up. And for the mice, the light at the end of the day is more important to extend. They need to extend their day a little bit each day to live on a true 24-hour period. We have to shorten the day a little from 24.2 to 24.
Yeah. And so I guess that's why, you hear a lot of people talk about today the importance of morning light for humans. You just basically told us the reason why. Because our period is a little bit longer than the 24-hour day, that the light at the beginning of the day is really critical. You see something a little bit different in other creatures like mice. So the details vary from species to species, but I think one thing that probably holds here is that the intrinsic loop that corresponds to this clock in our cells, it's not exactly 24 hours, and it has to be entrained to the external environment. So maybe one way to explain that would be, what would happen if we were just in a dark room? We wouldn't stay on the exact same sleep-wake cycle if we had no light to entrain to, right? We would kind of drift in one direction or another, depending on the species we're talking about.
We would definitely have strong circadian rhythms, but if my period is 24.2 hours, then each day I would be, on average, going to bed 0.2 hours later than I did yesterday, and again tomorrow, and again. And eventually those 0.2 each day would add up to a full day, and I would go around the clock. So this is the basis of those old cave experiments that many people know about, where people were kept in bunkers or caves where there was no light cue to the time of day. And we as humans then would drift later and later each day. Mice, because their periods shorter than 24 hours, if we turn the lights out in the vivarium and we measure their rest activity behavior, they advance a little each day and get up a little earlier each day. But the point is that you've made, which is absolutely correct, is that the period is about 24 hours and we're constantly adjusting it a little to live on the 24-hour day of planet Earth. Now, it's important to note, and we'll come to this later with regard to the paper that we will discuss, it's not so simple as every day being 12 hours of light and 12 hours of dark.
Right.
In the laboratory, we do that with mice. We keep them in a room where the lights are on for 12 hours and lights are off for 12 hours. That on planet Earth only occurs 2 times per year.
Mm-hmm.
And that's the equinoxes. Okay. But because of our relation to the sun and our orbit around the sun, the day lengths are changing in the summer hours and then changing again in the winter hours, with days becoming longer and longer, the photo period being longer and longer in summer, and then getting shorter and shorter in wintertime. And we're coming up to the December 21st, of course, which will be the shortest photo period of our days on planet Earth.
Mm-hmm.
And so not only do organisms running around on the planet have to adjust to, or have to be primed to function on an approximately 24-hour day and be able to adjust to a 24-hour day, but we have to do that in the context of the light changing, the length of the day changing every day.
Yeah. Yeah.
And around the equinox is when we have 12 hours of dark, 12 hours of light. If we're at the equator, it gets more complicated, because if you're living in northern climes like Scandinavia or far north Russia or China, the day, there are much longer days in the summer than we have here in San Francisco, and much shorter days in the winter than we have here in San Francisco.
Yeah. And of course, so no matter where you live or where a creature lives, if we're talking about a natural environment, so everything before, humans created the artificial environments we now live in, our bodies are clearly adapted to these natural seasonal changes in light. We can adjust pretty smoothly and easily to these incremental changes that happen day by day throughout the year. And obviously there's a lot of seasonal behavior changes that animals display to adapt to the changing light conditions of the natural world based on how the Earth rotates and is tilted and all that. But when you start to move in and out of light environments that are changing at a faster rate than that, eventually you can kind of completely disrupt the body's ability to smoothly transition. So I guess an example of that would be like when we travel, like I just did, and you get jet lag. You're basically moving. The light environment is changing faster than your clock can adjust to the new environment.
Right. And I've been traveling recently too. I just flew back from Austria on Sunday. So that's an even bigger time jump than yours to the East Coast. If I could come back to evolution for a moment, remember that evolution happens over very, very long time periods. We have not evolved to have a mechanism to adjust to jet lag.
Mm-hmm.
We do adjust. And as you suggested, it takes a little while to do that when you fly from New York to Seattle, for example. But that's using the very same mechanism that was evolved to allow us to adjust to the changing day length, and not to changes in the time zone of 9 hours that I experienced or 3 hours that you experienced. So that mechanism works for us, but it's not tuned to help us adjust quickly from a 9-hour time change. And that's why we have this thing we call jet lag.
Mm-hmm.
And some of us suffer more and some of us suffer less from that. But it's because we really haven't evolved to adjust to time changes of that magnitude. That's been a very recent thing introduced by flight and our ability to take jets across the ocean.
Yeah. And then of course, if we think about animals in natural habitats, especially those that are not right at the equator, where you've got clear seasons, spring, fall, winter, summer, you've got not only the light variation from season to season, the days get longer in the summer, the nights get longer in the winter, at least on this half of the earth. And, so there's that light that changes. Obviously, that light's going to go into your eyeball. It's going to convey information to the nervous system that's going to feed into these central regulators of our circadian rhythms in the hypothalamus and elsewhere. But there are also other environmental factors that change seasonally that are correlated with these light changes. And a big one is food and food availability. So what can you start to tell us here by way of background to set up this paper about how animals are also adapted to seasonal changes in food availability and how that ties into circadian regulation?
Okay, so let's picture ourselves as cavemen and women. And again, to survive, we have to live, we have to propagate our species, we have to survive, and we have to procreate. So as cavemen and women, I think it's reasonable to think about teleologically, really, what our bodies, our physiology is trying to do. There's going to be a lot more food and nutrient availability in the summer and fall, and much less in the spring and winter, or winter and spring. And so I think that the way I think about it simply in lay terms is that I believe we've evolved, and other organisms too, to be able to binge during times of plenty and put on fat that will serve us to get through the lean times.
Right. And we see this all the time, right? People can, the grizzly bears gorging on salmon and berries to get fat so they can survive the long winter, squirrels gathering nuts before they hibernate, and so on and so forth. So it should be pretty intuitive to everyone. There's times of feast and times of famine, basically. There's times of plenty, and animals are naturally, especially when they're in these environments where they've got seasons, they're not close to the equator, there's not always a stable food and light source. You're going to want to take advantage as much as possible of abundance when it's there, because it's not always going to be there. It's reliably unreliable.
And so if we just go back to the transcriptional, translational, autoregulatory feedback loop, it's like a ballet. These different protein levels rising and falling, transcription factors going up and down, regulating each other in such a way that the whole process in a single cell takes about 24 hours. That's happening in all of our nucleated cells. In addition, I should mention, by way of background, that you mentioned the hypothalamus, the suprachiasmatic nuclei, these nuclei in the hypothalamus, which kind of are the conductor of the whole symphony. If all of our cells are musicians playing instruments, we can all keep pretty good time. But if we don't hear each other, if I'm just playing my instrument alone, and you're playing yours, and we're both trying to play at the same tempo, when you put them together, it's not going to match perfectly because your tempo is not going to be exactly the same as mine. Mm-hmm. The SCN and the hypothalamus, this paired nuclear structure, is the central clock that is taking all kinds of cues from our biochemical milieu and then orchestrating the circadian rhythmicity of our liver and of our muscles and of our stomach.
It's sort of like the conductor in a symphony. It's keeping everyone in sync.
Exactly. And so problems arise, of course, when we get out of sync. So that is relevant to this discussion.
And so the hypothalamus is a key integrative structure in the brain. It integrates signals from many different parts of the brain, from all the different sensory modalities. It's paying attention to the light signals coming from the outside world as the seasons change and the days get shorter or longer that come in through the eye. It's paying attention to the food we're eating and its composition, which is going to vary systematically across the seasons, and so on and so forth. So the suprachiasmatic nucleus of the hypothalamus is like the central clock. It's the thing that's listening to all of that stuff and coordinating all of the clocks that are going in most of our cells. And we've got these systematic correlated changes in the external world, changes in light, changes in food availability, et cetera, the animals naturally obviously have to adapt to. And so the question is, how do we start to think mechanistically about how changes in things like light and changes in things like food actually like shift these clocks back and forth in different directions in a way that's adaptive for the organism? And so What else do you want to say here by way of background to sort of set up this paper? Is there any other key pieces of background information we should know?
Well, I think it's worth noting that the feast and famine, the one thing is constant, and for sure, and that's how fast the planet is spinning on its axis, right? So the total day length is constant, essentially. Um, Things like adjusting to light as you move to farther latitudes, north and south, that varies depending on where you are on the planet. So that's a whole nother level of complexity. Food availability, nutrient availability also is not the same this summer as it will be next summer, as it will be the next summer after that. And the length and hardship during the winter is different year to year. So to survive as a species, we have had to evolve in a way that we're able to adapt to the changes in food availability. But also, there has to be some flexibility with regard to just how bad the famine is and just how good the feast is. So I just want to emphasize that that's yet another layer of complexity. So it's really quite spectacular to think about. And we start with the basic transcriptional translational feedback loop, which is pretty easy to understand. Mm-hmm. But then the regulatory elements that are keeping that clock ticking in our brain and in our cells in sync with the environment that we're in because of how we've traveled north and south or not, and just how bad this summer and winter have been. There has to be flexibility for accommodating differences from year to year. And we have survived, so our genes have been selected for because we've survived better than some other, ancestors of ours who perhaps weren't able to adjust to the changes in feast and famine as well as we or our ancestors did.
And so when you understand some of the molecular biology here and you start to think, okay, so the 24-hour period of the day is constant. Every day is 24 hours. The light levels change throughout the season, but it's always 24 hours. The period of the molecular clock in our cells is also constant. It's close to 24 hours, not quite, but it's pretty close. So if we know that across the seasons, light levels change systematically, food availability changes systematically, and so forth, and you understand a little bit about molecular biology. So maybe people might understand, okay, well, you can turn up transcription a little bit, make more of a transcript and more of a protein. You might modify a protein in a particular biochemical way to make it last a little bit longer or a little bit shorter, make it a little bit more active or less active. You might phosphorylate it or dephosphorylate it. There's lots of biochemical moves that biology can make to shift some of these things. And so you might think, okay, a natural way I would imagine that the clock can shift to keep up with the changing seasons would be, okay, maybe there's some light dependent mechanism in how much protein gets made, or maybe as the seasons change and the composition of an animal's diet changes, maybe its fatty acid content changes, for example, maybe this affects how one of these transcription factors gets phosphorylated and it lasts a little bit longer as the fat levels change a little bit. So you can start to imagine how you would shift around this clock by playing with how active or how long some of these clock proteins last. And so do you want to start talking about this paper a little bit in terms of like how you guys set it up and how you started thinking about this and kind of the setup for why you started to do these experiments?
Well, I'd like to go back and set up a little more background. And so the very first family that got my colleagues and I thinking about circadian rhythms was this large Utah family. I was at the University of Utah at the time, a large Utah family that has in each generation a lot of people who are extreme morning larks. And so
they just naturally like to get up early. They're morning people.
Yes, but very extreme. 3 in the morning, 2 in the morning, 4 in the morning. And this is lifelong without an alarm clock. But they're so tired at dinner time when most of us are wide awake that they you know, are reluctant to go out to,
Yeah, it affects their social life and their ability to do business and socialize.
These people, most of them just live on their own schedule and they feel good about it. But occasionally they complain, because they can't stay up and go out to the late jazz show or whatever. They just simply can't stay awake. They're so tired. So this family we use to map and then clone the first human circadian rhythm gene, which is similar. It's a homologue of the Drosophila per gene, which was the first gene in Drosophila clone. And the particular amino acid residue, 1 in 1,000 residues that was mutated in the affected members of this family, is the change of a serine, which is a site of one of these biochemical modifications you mentioned, phosphorylation. And so we've done extensive work showing that phosphorylation of this site or not can tweak the length of the period. And in fact, the second gene we cloned is a kinase, which actually phosphorylates that very serine residue and others in the region, and is itself a circadian gene. So there's complexity that we don't want to get too much into.
Mm-hmm, mm-hmm.
But what's fascinating is that this residue is so critical to circadian timing, and we started studying it when we cloned the gene and published it in the Mutation 25 years ago. And yet there's still so much to learn, and it's very relevant to sensing the change in seasons as well.
Mm-hmm, mm-hmm.
So the circadian clock is a daily oscillation. But when we start to talk about seasons, we have to integrate daily changes over months
mm-hmm.
and up to a year to get through the whole spring, summer, autumn, and fall cycle. And it is that very same serine that we identified in the original family that is contributing to the ability of the clock to sense changes in the season, the day length, and to predict what's going to happen seasonally.
Mm-hmm.
And that in turn affects our the physiology of our storing fat and burning energy, fat to make energy to survive in these changing conditions.
Yeah, but at a high level, basically what you're saying is, using this genetic condition in this family in Utah as an example, if you get a mutation in the right gene, in this case, it's a gene that encodes a protein that's going to be part of this core molecular clock that's important for circadian rhythms. One sequence of DNA turns into a slightly different sequence. This then swaps out one amino acid residue for another amino acid in one part of one protein. And if it's a circadian protein that oscillates, as part of this core clock mechanism, and that new amino acid that you have because of that DNA mutation prevents or allows you to be phosphorylated or changed in some way, that will affect how long this protein lasts or where it goes, or some other aspect of its function or its timing.
Or how strongly it's able to feed back in the negative regulatory feedback loop that we talked about.
Yeah. But just a little change like that.
It's both stability of the protein, but also its activity in repressing its own transcription.
Yeah. And so it's a change like that results in a phenotype like this. In this case, their timing is different from normal people, and they're these extreme morning larks.
Right. So then the one other piece as background that I think is relevant is that my colleague Ying-Wei Fu published a paper in 2013 that I think is one of the most beautiful papers I've ever read. But it showed that the phosphorylation of the serines that are so critical to circadian and now we know seasonal timing, those very same serines can be modified in a different way by two enzymes, O-glucnac and O-glucnase, O-glucnac transferase. So serines can have a phosphate group, that's phosphorylation, and that can have regulatory roles as we've discussed. But this very same residue could also be glycosylated by adding a glucnac moiety, and the levels of glucnac in our cells are dependent on dietary intake.
Mm-hmm.
And so we had predicted way back in 2013 that different levels of glucose in the serum or glucnac availability to compete for modification of these serines, compete with the phosphorylation. It can be either phosphorylated or it can be glucnasylated. That would be a very direct tie of the core clock regulation to metabolism and food availability.
Mm-hmm. Yeah.
And so, in fact, it turns out to be true.
Yeah. And so, in the case of the genetics of the family that you mentioned, that had this morning lark phenotype, that's a genetic difference that's leading to a constant difference between that group of people and other people in terms of how these proteins behave and the timing of everything. But as we're getting to, you can also imagine that if the seasons change and the composition of the diet changes, maybe something in the diet results in a biochemical modification of some of these proteins. And as the diet changes seasonally, then the periodicity of this clock, this molecular clock changes, and that could be a way that these clocks shift over time in response to changes in food availability. So when you start to model this in the lab, these seasonal changes in light and or food, can you start to talk about how you guys actually did that in mice? I guess the obvious thing you can do with light is in the lab, you can just change the light conditions. You can go from 12 hours of light, 12 hours of dark to something else. So how do you actually model the seasonal changes in light and food in the lab?
Well, so first of all, we have wild type mice, which interestingly would never exist in the wild. They're laboratory mice, and for a number of reasons, their survival, they wouldn't have a survival advantage in the wild, but they work very well for the kind of experiments we're doing. The first thing we can do is take a wild type mice, a control mouse that has the normal mouse sequence for this particular gene. And then in another line, we can engineer the mutation that we found in the humans. And what phosphorylation does to the protein is to add a positively charged moiety to the amino acid. And so we also said, well, what happens if we change? So serine doesn't have a charge. The amino acid doesn't have a charge. If it's phosphorylated, it has a negative charge.
Mm-hmm.
I'm sorry, a positive charge.
Yeah, positive charge.
So it's the charge on that amino acid that's causing really big differences in the ability of, or the length of the periodicity of the system.
Mm-hmm.
So we didn't find this mutation in humans, but we said the woman who was working on this, the postdoc, Xuing, said, well, I'm also going to engineer a mouse that has a negatively charged amino acid where that serine normally is. So now we have 3 different mouse lines, the wild type line that has a serine at that residue, the mutant, which has a glycine, just like the humans, which cannot be phosphorylated. And so it's always got a neutral charge as an amino acid. The serine is somewhere between uncharged and plus charge, depending on how much of the protein is phosphorylated at that site. And then we have a third model or third line where there's a negative charge there.
Right. So you've got the normal mice, and then you can engineer another type of mouse that mimics the human genetic condition we spoke about. And then you can make yet another type of mouse that sort of has, from a biochemical standpoint, the opposite or distinct type of biochemical change. And you can ask, how do all of these 3 compare to each other in terms of their periodicity?
And so what we showed, way back in 2007, is that one amino acid, if it's regulating its charge from 0 to -1 or +1, that the changes in the charge at that one amino acid can change the periodicity of the clock by more than 4 hours.
Which is huge.
Huge. And so again, just to get back to evolution for a moment, these families have not been selected for this mutation. If there was selection, then many more people would have this mutation. But they've not been strongly selected against, and so they have survived. So this is a mutation that hasn't interfered with human survival, but I don't think that it probably has a particular benefit or risk in terms of survival on our planet. And so I've kind of lost my sight of the point I was trying to make. But the charge that's regulated by these biochemical changes of the protein can dial the period of the system from 20 hours to 25, 26 hours.
Yeah. So whether or not you've got one amino acid in one part of one of these key proteins can have a huge effect on things like this. And you guys can genetically engineer these things in the lab in mice. There are certain families, like the one you mentioned, that might have a genetic mutation that makes them this way. But then you can also start to think, okay, well, even if you don't have a genetic mutation at this residue, environmental conditions might change, lighter food levels, say, such that these biochemical modifications at this residue, at the normal protein, might happen more or less. And now you start thinking like, oh, okay, this could be a way that things are biochemically regulated to shift the clock in response to changing environmental conditions season by season.
Right. And because we know that phosphorylation is so important, regarding the charge on that residue, we predicted that if there are high glucose levels and therefore higher glucnac levels in cells, that this competing enzyme system, O-glucnac transferase and O-glucnase, could compete for occupancy at the serine with an uncharged residue versus the kinases and phosphatases, which are trying to put on a positive charge. Mm-hmm. So that is the tie to metabolism. And we've shown that in diabetic mice with poorly controlled glucose, that their period is shorter. And it phenocopies. It phenocopies the human condition, which we call familial advanced sleep phase, FASP. Mm-hmm. Hyperglycemia in a diabetic mouse model phenocopies FASP because it leads to hypophosphorylation of this serine and other serines that are downstream. So again, some mechanistic insight into how the molecules that are involved in connecting seasonality, sensing of seasonality, and food abundance.
Yeah. And so everyone will probably find it pretty intuitive that food, even just the amount of food you eat or whether or not you're eating can have an effect on your sleep-wake patterns. A lot of people find it difficult to go to bed hungry. You can eat a big meal and it makes you tired and so on and so forth. So food definitely ties into sleep-wake patterns and activity patterns in a big way. Can you start to talk about this paper a little bit? And, maybe one place to start is just when you're studying circadian rhythms and periodic behavior in mice in the lab, how do things like calorie restriction and going to a high-fat diet from a normal diet start to affect circadian entrainment to the external light cycle?
Yeah, so this was motivated by a project started by a postdoc, Dan Levine, who's interested in metabolism and metabolic links to the clock. And so he began by doing an experiment where he moved the wild type mice, the normal mouse sequence, the mutant FASP model mouse, and the one with the opposite change that introduces a negative charge. Those three different mouse lines, he put them into cages where he can control their feeding, how the food is distributed over the day and how much food is distributed, calorie restriction or not, and ask the question, when he changed the mouse environment from a 12-hour light, 12-hour dark equinox kind of situation, to a winter schedule, and he chose 4 hours of light and 20 hours of dark as an extreme winter schedule.
So you got 12-12 light dark, sort of like middle of the summertime. It's bright for a lot of the day, and then you go to a 4-hour light, 20-hour dark schedule. So it's like deep in the wintertime at a fairly northern latitude.
Right. And then the opposite. If you go from a 12-12 timing to a 20 hours of light and 4 hours of dark, which is much more like summertime in Scandinavia, or wintertime in the southern hemisphere. I guess that would be southern. It would be summertime in the southern hemisphere, which is at our winter time.
Yeah.
Sorry for the confusion. So what he saw that was really striking was that he could affect the ability of so this is like a jet lag experiment.
Yeah.
Sort of. It's a little different, but it's similar to a jet lag experiment. Changing the light schedule, and we don't do it over many days or months of migration. We do it overnight.
I see. So like one day the light just goes from 12-12 to 4:20.
Right, or 24, and that had dramatic effects on how the mice were able to entrain or get back on track to the new light schedule. Normal mice on planet Earth don't have to worry about things changing overnight like that. This is an experimental paradigm in a laboratory, but it's very telling because it goes from the equinox to a very winter extreme or a very summer extreme.
So the question is really, how quickly do the mice change, and how does the dietary condition affect how quickly they change?
Right.
And so what's the basic answer here? When mice are food restricted or they're on a higher fat diet, how does that affect how quickly they adapt to the new light dark cycle?
I'm going to grab the manuscript. So take a short break here. 1 second.
Okay. Yeah, we're talking about a paper that I'll link to. Just came out a little while ago, but it's called Unsaturated Fat Alters Clock Phosphorylation to Align Rhythms to the Season in Mice. And there's a lot going on in it. So he's grabbing the paper just to make sure we have all the details. But it's all about how the changes in light cycle and changes in dietary fat in particular affect the ability of animals to entrain to these light cycles.
Okay. So, for example, when the mice that had the human FASP mutation were changed from a light dark 12-12 to a winter schedule, they were able to entrain very quickly, whereas when they went in the other direction, from a 12-12 to a summer schedule, they had difficulty in entraining to the clock. So the food availability or the light schedule affected their ability to adjust to the seasonal change, which we interpret to mean that less availability of food in the wintertime makes it possible to advance the clock more quickly, and the opposite is true for food abundance in the summer.
Got it. So whether or not you've got more food abundance or you've got more food scarcity, this basically affects how quickly the animals adapt to changing light conditions.
Right. And again, to emphasize, we're looking at a very dramatic change that happens overnight by just changing the light schedule. But what's really happening in the world is that these things are changing more slowly.
Right.
The lengthening of the days is not constant across years, but when it's changing most quickly, the length of the day may be shortening or lengthening by as many as a minute or two per day. But at the equinoxes, it's coming the amount of change per day is at zero, and then it begins to go in the opposite direction, right? So at the equinox, we're 12-12. And if we're moving into the winter time, the dark period is getting longer and longer and longer, and it's getting longer faster at the solstice. And then slower and slower and slower as we come back to the equinox on the other side of the year. So the point is that we're making this dramatic change, which is unnatural.
Right.
And seeing how the mice adapt to that.
Yep.
Their ability to re-entrain to the new schedule.
Yeah. And I guess, so you guys notice, like, okay, if mice don't have access to much food, or they've got more food, or basically higher fat content in the food, this is affecting how quickly they adapt to these sudden and dramatic changes in light. You might start to think at this point, okay, well, something about these conditions must be affecting the core clock proteins or some key components of the whole clock apparatus. One thing you might think is, okay, well, maybe some of these things, maybe some of the dietary components or whatever, maybe they're affecting the phosphorylation of these key transcription factor proteins, or how much of the protein is there. And therefore, this can extend or truncate some of these transcription translation loops that happen within cells. What did you guys start to uncover about how, say, a high fat diet compared to a normal lab mouse diet was affecting the components of the key molecular clock inside of cells?
Right. So I think to go over all the experiments would be too complicated, but the basic idea that Dan had is that he wanted to do metabolomics. So we have the ability to measure metabolites in the tissues of mice. And he did this in mice with the different mutations, but also under the different conditions of calorie abundance. Calorie abundance is that we give as much food as the mouse wants to eat.
Yep.
And they do that. Calorie restriction, of course, we have an apparatus which will distribute a food pellet every so often, and we can adjust, how many food pellets the animal gets over the course of 24 hours, and the timing during the day when he or she receives the food. And that is an experimental condition. We can see what changed. And what Dan found was that some of these fatty acids were dramatically changed under conditions and it was parsimonious in that the winter condition looked the same as the calorie restricted condition.
And when you say you did metabolomics, are you looking in a particular tissue here? So my understanding is you're looking at the hypothalamus. So you're kind of looking in the region of the brain where this core conductor of the central clock lives. And when you say metabolomics, you basically mean you can do more or less an unbiased test to see how all, or at least many of the metabolites in this tissue in the hypothalamus are changing. So you can take a pretty wide and pretty unbiased look at everything that is changing in this key regulator of circadian rhythms in the hypothalamus.
Exactly. And so I don't remember the exact order, but you can do it in the 3 different lines of mice that we have. The FASP mouse, the wild type mouse, and the one that has the amino acid that's negatively charged. And there were dramatic differences, particularly in fatty acids, that could be measured when he isolated hypothalami from those mice, ground it up, and sent it off to a metabolomics lab where they use sophisticated techniques to look for, certainly not all, but many, many metabolites that we have in our normal cells. And he found that the most dramatic differences between the 3 different mutant lines. Let's talk about the FASP mutant line, which is the one that has a human mutation, and it has a shorter period, circadian period, but it also adjusts in a way that's consistent with food availability. And so the summertime schedule yielded similar results to what we saw in the mice that had the human FASP mutation.
Mm-hmm.
On the converse, the winter cycle, the changes in the winter cycle were consistent with what we saw in the mouse that carried the negative charge at the serine, consistent with it not being able to be oglucinacylated and for phosphorylation to be more prevalent.
Mm-hmm. So you've got these three mouse lines. You've got these different conditions where you can change the light cycle suddenly. The rate at which they adapt differs from mouse line to mouse line. And you've got some pretty sophisticated, unbiased techniques that allow you to do metabolomics in the hypothalamus. So in other words, you can see what's changing inside of cells in terms of all the molecules and stuff that might be changing inside of these master regulators in the hypothalamus of the circadian rhythm. And basically what you find, so you could have found, anything. You could have found, X, Y, or Z going up and down. One of the things it looks like you guys zeroed in on is that some of the biggest changes were seen in polyunsaturated fatty acid metabolites. And so what can you start to tell us there in terms of what the metabolomics experiments were showing you? What were some of these families of compounds or metabolites that have to do with polyunsaturated fatty acids? How are they different here, and like what were they, and why do you think these things were what jumped out of this experiment?
Yeah, so I'm just looking at some notes I've made here because it's very complicated. The key thought is that in non-thermoregulatory organisms like cyanobacteria and plants, increased polyunsaturated fatty acid production during the winter is part of an adaptation to survive cold temperatures.
Yeah.
Thus, the animals have access to PUFAs during the winter.
Yeah. This would be like why like a cold water fish, like a salmon, is kind of famous for having high levels of omega-3 polyunsaturated fatty acids. The idea is it's cold-blooded, it lives in cold water, and it needs to have these PUFAs because they help it not to turn into a fish stick. It helps keep the fluidity of the membranes.
And so it becomes confusing in our experiments with high-fat diet, where there's high fat content, but also a high monounsaturated fatty acid versus polyunsaturated fatty acid ratio, making it mimic a summer MUFA-PUFA ratio condition, if you will. So it's important, I think, to focus on the monounsaturated and the polyunsaturated ratio in these experiments.
Yeah, and I think that's important. I mean, there's a number of reasons why I think that's important. So MUFAs and PUFAs, two different types of unsaturated fatty acids. They do different things. They can metabolize into different things, and they often sort of compete in a way for access to different enzymes and stuff. So the balance of these things is critical for a lot of biology. So you would imagine, in other words, at a high level, that just changing the MUFA-PUFA ratio is going to affect all sorts of things in the animal cells.
Right. And in winter, animals have more access to polyunsaturated fatty acids in their diet. And we show that the diet low in polyunsaturated fatty acid enhanced the animal's entrainment to a longer summer-like schedule, but decreased their rate of entrainment to a winter light cycle.
And so one of the things that's really interesting here is that a lot of the metabolites of polyunsaturated fatty acids are things like, there's all sorts of things, but there's inflammatory compounds like prostaglandins. There's also endogenous cannabinoids like anandamide. And these things are famous for regulating feeding behavior and food intake and food palatability and so forth. And so now it starts to make sense for why some of these things would be regulated by food restriction and high fat diet, and would vary in this type of way because right, a mechanism in the brain for stimulating food intake and getting animals to eat more, if they wanted to say prepare for winter, would be increasing things like endocannabinoids. So it starts to sort of make sense when you think about why polyunsaturated fatty acid biology would be so sensitive to these feeding conditions, because a lot of the metabolites of these dietary PUFAs are themselves going to be involved in, regulating things like food intake and adiposity and so forth.
Right. And, as we began in the background, just talking about, the core circadian clock, and then slightly more distal regulatory elements like phosphorylation, glycosylation, and the downstream effects of those changes in affecting, downstream transcription factors. There's immense complexity. And so we're studying one amino acid, and the charge on that one amino acid out of 1,000, or approximately 1,000 in the PER2 protein. And this is clearly important in the regulation of the seasonal adaptations of the clock, or the adaptations of the clock to the seasonal changes, I should say. And yet there are also plenty of other regulatory things going on. But we're trying to get our hands. So Dan was trying to get his hand on some molecular pathway that was very direct, where there was regulation. So that's what this most recent paper shows. In addition to what's reported in this paper, there's also many other regulatory things going on in this 1,000 amino acid protein that are contributing to regulation as well. This is one that's very important to seasonal regulation, but the complexity I can't emphasize enough is much, much greater than we can even begin to imagine at this point. Mm-hmm. And we're trying to focus on finger holds and toe holds that will allow us to get deeper and deeper into the overall regulation.
One of the interesting things you did to investigate this question of the MUFA-PUFA ratio and how it affects things is you actually have what you call the high-fat diet, and then you've got another high-fat diet, which involves partial hydrogenation of the dietary fat. And basically what you did is you were able to alter the MUFA-PUFA ratio, but hold the total percentage of calories from fat and the calorie density constant. So you got two diets that are essentially the same, except one has got a higher MUFA-PUFA ratio and one's got a lower ratio. So when you manipulated that as a variable, the monounsaturated fatty acid versus the polyunsaturated fatty acid content, how did that affect things here?
So increasing the ratio led to a tendency to be more winter-like, and the opposite, changing the ratio in the opposite direction, had the effect of producing a more summer-like behavior.
Got it. And so the idea here is that in natural habitats, there's probably going to be a natural shift in the balance of mono and polyunsaturated fatty acids that are just out in the world and available. And as these things shift seasonally, this is going to sort of help facilitate the shift back and forth between the winter activity pattern, the long night activity pattern, and the long day activity pattern. And this has to do with the balance of MUFAs and PUFAs dictating how readily different metabolites get produced, right? So you might get more of one set of metabolites when the PUFA levels are higher, another set of metabolites going higher when the MUFAs are higher. And at least some of these things are coming in and affecting the period proteins and the components of the clock by, facilitating their phosphorylation or dephosphorylation or whatever. But I guess the point is, at a high level, the balance of dietary fatty acids, in particular, MUFAs versus PUFAs, dictates how quickly the molecular clock can shift, because it's ultimately going to affect some of these key proteins that are part of this circadian, molecular loop.
Right. And, we began talking about the simple negative autoregulatory loop, the transcriptional translational feedback loop, which is pretty simple to understand. I mean, and we can draw models that look, say, well, that makes a lot of sense. But the complexity then comes in with all of these different modifications. And then once you change the circadian output, some of those genes are transcription factors themselves. And as you mentioned earlier, they regulate transcription of other genes downstream that are relevant to physiology of all sorts. Some of the circadian transcription factors regulate the transcription of other transcription factors, which have downstream effects on yet other genes. And so this is why, it's so difficult to imagine, how the system can work. To me, it's so immensely complicated, but it has been evolution that's allowed, for the selection of the things that work to lead to survival and propagation of the species on a planet with this day length and these kinds of light and dark conditions.
One of the things that's interesting to start to think about here is when we think about the modern human environment and how it's different from natural environments and how it ties into some of the things that you guys observed in this paper. So in particular, when I think about humans today, like you and me, modern humans living in our technologically advanced society, on the light side, we've got more light than ever, right? We've got all of these LEDs, all of these electronics. They're beaming blue light, which is essentially like daytime light to our brains, telling us that it's daylight more often, right? When the sun goes down, it's no longer pitch black outside and we have nothing but, torches and fires. Now we've got iPhones and computers, and all of the lighting in my home is, so it's in general, directionally, our brains are being told the days are longer or it's still daytime more than the real daytime. On the food side, what's really interesting is one of the things that characterizes the abundance of processed foods is the high polyunsaturated fatty acid content of the diets. So modern humans today are living in a more daytime light environment, more light, brighter light, more hours per day. And we have more polyunsaturated fatty acids in our diet and in our bodies than probably any time in human history. And so what do you start to think about there in terms of how those two features of the modern human environment are playing into our circadian rhythms?
Wow, big question. Yeah, what you said is exactly right, and it was well said. The other thing to note is that we have supermarkets that are open 24 hours a day, so food availability is, and fast food joints where we can get huge meals with huge amounts of PUFAs and calories. So these have all been relatively recent, since the invention of light. If we think about going camping, when we camp, we live more like the cavemen and women did, because the sun goes down and we don't have light, and we might build a bonfire and sit around it and roast marshmallows. But when I'm camping, I go to sleep pretty early and I wake up when the sun comes up, right? That's more what the cavemen and women tended to do. In addition, other things that regulate the clock, you've mentioned food availability, but also exercise. Cavemen and women ran around a lot. They were hunter-gatherers. Some of us in modern society are more sedentary. Some of us, exercise every day. But all of these things are feeding into the circadian regulation. But what's happening with the invention of electric lights, we're getting way more light at times that we never got light as cavemen and women. And we can go to the refrigerator and grab food in the middle of the night, or a chocolate bar, as I am sometimes guilty of doing, because I can't sleep. And so we've changed the environment that we live in so radically over a couple of hundred years. And more recently, with all of the cell phones and pads and computers and so forth, we're getting light at very unnatural times of day. And I think that all of these things are contributing to us, or to our epidemic in this country of obesity, which is a huge problem and which contributes to the increased risk of diabetes, and the incidence and prevalence of diabetes has been going up and up and up in our country and many other developed countries. So this has real implications for our health and happiness, I think. And so technology is a great thing. I like having electric lights, but I think it's also important for us to think about trying to live in harmony with a more natural environment.
Mhm.
And so I do encourage people not to take their phones to bed, not to take their iPads or other, electronic instruments to bed with them, and to try to get a good night's sleep, because that's so critically important for our general health.
Yeah. One thing I think we maybe forgot to emphasize earlier for people is that we see the core molecular clock that we see is highly conserved across animal species. A lot of the same components are in mice and humans. They're even in Drosophila, like fruit flies. So a lot of and you can imagine why, like every animal lives on this earth and we all have to adapt to the same, solar cycles and light cycles. So it makes sense there's going to be a high degree of evolutionary conservation there. There's also going to be differences. Obviously, mice and rats tend to be nocturnal. We're diurnal. There's going to be differences in the details, but this core clock has the sort of same structure and logic to it across species. And so you would expect that a lot of the results that people like you and others in the lab see in these creatures are going to apply to humans, at least directionally or according to the same principles. One of the results that we talked about from you guys is simply that if you change something like dietary fat content, even not just the total amount of fat, but the MUFA-PUFA ratio, that alone could be sufficient to shift how quickly your body can entrain to external changes in light and other things. So that would imply that if people just shifted, say, the MUFA-PUFA ratio of their diet, that alone could have some effect on how their circadian biology is working, how readily they're adapting to changes in seasons and light levels and things like that. Is that a fair statement?
Absolutely. But because of the complexity of the system, I don't feel comfortable saying, there's so many fad diets of different sorts to help, with different things. I think we still don't understand nearly enough about the complexity of the system to make strong recommendations. But in general, I think, trying to live like cavemen and women in the sense that we eat, nuts and berries, vegetables, also some meat, but that we eat during the light phase. I sometimes find myself going out to dinner late at night, and this is not what the cavemen and women did, and that it's not healthy to go to bed with a full stomach. I don't sleep as well. So I think eating our meals during the physiologically correct time of day, which would be the daylight hours, is, I think, something that we should strive to do more. Will I have a pizza late at night at a party, or playing board games with friends? Sure, but in general, I think it's better to focus on getting our main caloric intake during the light hours, and not so much just before bedtime, so that we can sleep more deeply, as I do when I don't have a full stomach.
Yeah. Do any of your results imply things that might help people, say, adjust to new time zones? So if you've got to go to Austria for a conference, or you go on vacation several hours away, in a brand new time zone, and you're jet lagged, and you want to shift your clock as quickly as possible to adjust, is there anything implied by your results in terms of like, oh, you should calorie restrict, or you should fast for a day, and that'll actually help you adjust faster?
Yeah, so I flew not 9 time zones east. I flew into Munich and then took a train into Austria. But I would predict that eating lots of Apfelstrudel and Zuckertorte would help me align to the new time zone, because it's phenocopying, as I said before, the effects of the FASP mutation in terms of our ability to adjust our circadian cycle. I'm not talking about seasonal cycles. I'm talking about the jet lag kind of problem.
Mhm.
Now, of course, I enjoyed a lot of very good apple strudel, but it's also true that eating too much apple strudel is not that good for other elements of my health, like my weight. And so there's the issue of trying to tweak our system to adjust circadian time to jet lag. But there are also all the other considerations. I still don't want to gain weight. I still want to exercise and be very active, get a lot of light during the day when I'm out walking around Vienna or Salzburg. Um, and and so yeah, I don't think that this is not something I did intentionally. I was eating apple strudel because I really love a good apple strudel, but I wouldn't recommend that as a way to adjust to jet lag, even though theoretically I think it would help.
Mhm. What sort of experiments are you guys doing? Any follow up experiments on this, or anything to build on this general area of how changes in external factors like changing light levels, like changing food composition, affects the whole circadian apparatus?
Yeah, this is not an area that I plan on pursuing. Dan is an outstanding postdoc, and he really brought the metabolic expertise to my lab. But this is something he's now out on the job market, and so this is something that he very much plans to pursue in his own independent lab and as a young investigator. Um, so this is a huge area of interest and importance,
Mhm.
but it hasn't been a primary focus of my work, and therefore I'm encouraging Dan to do this in his own laboratory as he
Yeah, yeah,
starts off in his independent career. But yes, there are lots of experiments to do and trying to understand more details about the regulation and what is downstream of the PUFA-MUFA ratio that's leading to the ability to entrain to the seasonal changes. That there's still many, many unanswered questions that will be fodder
yeah.
for further work in his lab and in the lab of other investigators who read this paper and who have expertise in this area.
Yeah, I guess one of the big takeaways for this paper for me is that pretty intuitive and unsurprising, I think, to most people, that, right, obviously changes in light are going to affect your sleep wake cycles and your circadian rhythms and stuff. But even just independent of that, even just changes in the composition of your diet can start to shift that stuff as well. And to piggyback on something you said earlier, about, trying to live like the cave people, so to speak. Um, One thing that strikes me here is that is the idea that seasonality in food really matters. It used to be, and it is for most animals in the wild, that food is seasonal. An animal can't control what's available and what's not in the winter versus the summer, and its diet is going to reflect what's available in the natural habitat. Because of the modern human environment, the fact that we've got all of these packaged foods, we've got grocery stores or whatever, we've sort of, I mean, there's some seasonality. Like, obviously, sometimes we eat more pumpkin pie during certain holidays and other things, but we've kind of erased or minimized seasonality, I think, in food, because we kind of have access to all the food all the time now. And that could be worth thinking about eating a little bit more seasonally, because probably for most humans and for most of our evolutionary history, just like for most animals in the wild, these fat ratios, these nutrient ratios of all kinds would have been changing systematically season by season, and correlating closely with changes in light levels and all the rest. And we've largely broken that, I think, today with the modern food environment. We're sort of eating everything all year long.
Right. Absolutely. And again, evolution happens over very long time periods, but our diets today and our 24/7 kind of culture will have some selective disadvantages. So over hundreds of thousands or millions of years, some of the things we're doing now would be selected against. Uh, but in general, I think, we're not going to change evolution by our behavior. Evolution happens despite our behavior. But living in harmony with our biology, I think, is a really important thing. And, there are these extreme normal people with FASP who wake up really early, go to sleep really early. We have families with familial delayed sleep phase, which means that they're extreme night owls. They can't go to sleep before 2 or 3 in the morning, and then they want to stay in bed till later. To the extent that we can live in harmony with what our biology and our genetics is telling each of us, we're going to be healthier and happier.
Yeah.
The delayed sleep phase is more of a problem in our culture because it doesn't fit as well with the 8 or 9 o'clock school and work start times. And so people with familial delayed sleep phase, or even sporadic, the much more common night owls that are sporadic and not genetic, that is a harder that's a harder regimen to live with on our planet, in our society, based on, societal norms for work and school start times, for example. But other people say, we know that there's some people who can sleep with a lot fewer hours for their whole lifetime and still be healthy and happy. We know that there's some people who really require 11 or 12 hours of sleep to be healthy. But most of us need 8 to 8.5 hours. There are some folks who say, oh, everybody has to sleep 8 or 8.5 hours, or it's going to be really bad for you and your health. That's simply not true. For some people, they only need 4 hours, and that's all they need to be happy and healthy. But for others, they may need 12 hours because of the normal genetic variance that happens in populations. And so being mindful of what my body is telling me, if I am a long sleeper and I need more sleep, getting the best quality sleep for as long as I can is going to be, advantageous for me in terms of how I feel and how good my health is over my lifespan. So we have to be sensitive to the differences from, and I'm not saying we stigmatize any particular trait, but we have to be mindful of what each of our own individual traits are based on the genetics. And then, of course, everything is modified by the environment, the lights, the caffeine, the alcohol, the medications we take, the exercise we do or do not get. We have our biology and our genetics, and that interacts very strongly with the environment. So to the extent that we can provide a healthy environment, good quality sleep for as long as we need, as I need or you need, and having, a healthy diet and exercising regularly, all of these things clearly are beneficial to allowing us to be happy and healthy for longer as we as a species live, are tending to live longer and longer.
Well, I think that's a great place to end it. Once again, thank you very much for your time. I'm going to link to the paper that we discussed today in the episode description if people want to check it out. But if there's nothing else, I think that's it. Thank you.
Thank you so much. I really enjoyed it.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about that aren't necessarily on the box? This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast, and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.